Good day. Welcome to the Waters and TA Instruments medical device series. This series aims to show you how we can help you understand your medical devices better from a chemical and materials characterization perspective. This can then be applied across your workflow, from product development to processing to regulations or even troubleshooting, presented each week by an expert in the respective technology. For this webinar, we encourage questions, and we'll have some experts on hand to answer as many as we can in the time we have. Those we can't get to today, we will follow up with afterwards. Please post questions in the top left window and we'll answer at the end of the webinar. There are links to resources at the bottom window and a survey in the top right which we appreciate you completing. I would also invite you to register for future episodes of this series. This series will comprise of multiple sessions, one a week. These will cover various aspects of medical devices and the materials used for these devices. And we'll highlight how some of the technology Waters and TA Instruments have can answer some of the important questions that you have. Today's talk is the chemical characterization of medical devices. I'm Ben McCree, responsible for the medical device market at Waters. And I'll be your host today and for each session of the series. The main speaker today, after my introduction, will be Dr. Jing Lin. Jing has broad and deep knowledge of analytical technologies, in particular chromatography and mass spectrometry, and over 20 years of industrial experience in various markets. In her current role, she strives to develop the strategic business of material sciences within Waters TA Instruments and to enable the implementation of innovative solutions through keen understanding of industrial trends, customer needs and challenges, and effective collaborations with key opinion leaders. Welcome to the opening talk of Waters and TA Instruments medical device series. We start with an introduction to medical device characterization. Medical devices are essential for the well-being of millions of patients worldwide. From adhesive bandages to heart valves, medical devices are crucial in diagnosing, prevention, monitoring and treating illnesses and overcoming disabilities. Material development is at the heart of this industry, whether to increase efficacy and reliability, reduce costs, or enable personalized solutions. For safety reasons, there are constraints on what material can be used, but medical device manufacturers, enabled by the value chain, are pushing technology forwards. Ensuring safety on a product with a potentially quite complex build, different materials, different supplies, can be quite challenging. As the regulatory framework becomes more stringent, manufacturers need to marry physical performance with the intended use cases and expected lifetime of the device, ensuring that nothing comes off these devices that could be harmful to a patient. These chemical material properties are critical to the product efficacy, product safety, and ultimately the product success. To exemplify, if we look at a simplified value chain for the manufacturing process of plastics, there are several steps along the way, from monomer to the final product. Each of these steps can intentionally introduce additional chemical additives, but also non-intentionally added impurities and degradation products, which must be removed or characterized. Many of the issues arise due to the manufacturers not being aware of the impurities in their raw material at the levels that are critical for medical device regulations. Those that are used to other end markets might not have tight enough control on impurities, nor the batch-to-batch consistency. Historically, many of these companies were loath to disclose any information regarding chemical composition, the formulation of the plastic. It's usually proprietary information. Thus, the collaboration between manufacturer of the material and the medical device manufacturer must be close to ensure a smooth route to market and the creation of a safe and successful device. This is being reflected in the regulations. For instance, the biggest change for the EU market is the creation of a regulation which is combining two directives from almost 30 years ago. This is a very significant strengthening of the regulatory framework. There's an awful lot to unpack within these regulations, but for the purpose of chemical and material characterization, some of the items of note include the greater inclusion of the value chain and the inclusion of some devices which weren't covered before, that weren't considered medical devices. So for example, it's not only the site-corrected contact lenses that are included, but also ones that are only used for cosmetic reasons, to impart colour for instance. The suite of standards known as ISO 10993, the biological evaluation of medical devices, has also seen major changes. It's encouraging a risk management approach. To perform a biological evaluation of a device, your device's intended use guides the line of thinking. The nature of the body contact. Is it in contact with external skin, or is it implanted? The contact duration, is it only used for an hour, or can it maintain contact for weeks? Historically, this was used to dictate the biological testing. The new standard appreciates that if you know the material formulation, if you understand the chemistry, then you can predict the risks. So it's inserted chemical and physical information as the key step to intelligently design a biological evaluation. If we focus on the chemicals more than materials, we see the importance of contaminants, residues, all of these potential moieties that have been added either intentionally or non-intentionally. So the burden of characterization is increasing, but so is the recognition that the value chain needs to do more. These devices can be so complex that by the time the end manufacturer is testing for everything there might be, critical things could be missed, increasing the risk to patient safety. The value chain working together will alleviate many of these risks. Indeed, identifying a contaminant at the stage it was introduced greatly reduces wasted time chasing it down at a later date. There are many parts to ISO 10993. For the chemical characterization, here are some of the most pertinent ones, ignoring the degradation products for now. Part 1 describes what is a medical device. Part 12 describes extraction conditions, so the temperatures, times, solvents. Part 17 describes the limits for chemicals that might leach out of the compounds to the patient and how these leachables are set. And Part 18 describes the analytical techniques that can be used to understand the chemical and material properties, but today's talk will be focused on the extractables and leachables components. Taking the workflow from ISO 10993 Part 18, the first step is to gather information on the materials and chemical constituents. Depending on how much information you have from your supplier and the device's intended use, it is possible to come to a decision on the biological testing at this point. If there is not enough information, or the risk is not acceptable, then an extractables testing is required. Extractables are the potential impact of the device. Conditions are used that are beyond what the device is expected to experience to draw the chemical moieties out of the material. Alongside extractables, we have leachables. These represent the actual impact, what substances are released from the material during its clinical use. So first of all, we need to calculate what is known as the AET, the Analytical Evaluation Threshold. This is a new concept introduced at the start of this year in ISO 10993 Part 18. Below the calculated threshold, any extractables or leachables need not be identified nor quantified. That means, of course, that above this threshold, you need to identify the substance, you need to quantify this substance. So, you want to perform the study, you're going to perform the extractions. I will mention at this point that there are various extraction methodologies depending on the risk factors I described earlier. It makes sense that if a device will remain within the body for an extended period, that's going to have an exhaustive extraction. Whereas one that has limited contact can be treated under simulated clinical conditions. So you have this range from simulated to exaggerated to exhaustive. At this point, you might have enough information to understand the risks and come to a conclusion. But you might not. Depending on what the extractables are, now all identified if they're above the AET, additional characterization could be required here. This is leachable testing. So leachables are the actual impact. So simulated extraction is used to produce a profile which represents the worst case leachables profile. So targeting the leachables that you've identified in the extractable studies, under a more clinical use environment, you have the information to understand the risks and can plan accordingly. Looking at the workflow for extractables from an analyst perspective, once they've collected their data, they need to establish if anything is above the AET, the threshold which dictates identification and quantitation of peaks. For anything above the AET, the analyst has to identify. Having a list of potential candidates from the value chain would greatly simplify this process. If it is a complete unknown, then elucidation tools are required to confirm the structure. Once this is completed, it can be quantified. If the extra leachable steps is required, then the process is more targeted. A list of compounds from the extractable study is identified for targeted quantitation. This has less demands on the technology and software than elucidation as shown in the previous slide. To go through this in greater detail, I will hand over to my colleague Jing Lin. Jing.
Thank you, Ben, for this nice regulation overview and the introduction. Now let's talk about how Waters can help in extractable and leachable testing. Waters has a focused, well-integrated, end-to-end solution in this application area. It encompasses our core competency in chemistry, separation, mass spectrometry, informatics, reagents, and standards. Additionally, it is supported by a global service and application support capability to ensure customer satisfaction and success. During my part of the talk, I will give you a brief introduction to some of the key technologies, explaining how they help overcome the most common challenges that Ben laid out in the previous slide. We have two high-resolution MS solutions to offer for E&L studies. First, the Xevo G2XS QTOF for routine analysis of E&L studies. Second, the Van IMS QTOF provides extra capability with ion mobility for more advanced analysis. Many of you are probably familiar with the use of nominal mass and tandem quad technology for screening. However, high-resolution accurate mass technology is becoming increasingly attractive during the past few years. For targeted screening, its sensitivity now rivals triple quad systems with a far better specificity. But more importantly, we can get very good information about unknown peaks found in the samples, which is arguably the hardest part of the work. Furthermore, the data acquisition uses an easy-to-set-up generic method for both targeted and untargeted screening. Those data are particularly interesting for retrospective analysis. We can always go back to the data and see if compounds that we were not suspecting may have been present in earlier samples, as oftentimes we simply don't know what questions we want to ask at the time of data collection. Now, a little bit more information on exact mass for those who are not very familiar. Every chemical element has a nominal and exact mass as shown here with the examples of carbon, hydrogen, and oxygen. A quotable instrument can only measure the nominal mass of molecules with typically 1 decimal place. The example here shows an ion detected at m/z of 311.1. With a tolerance of 50 ppm around this nominal mass, this would result in 37 possible chemical formulas. That is a lot of possibilities to go through to come up with a final structure for this unknown compound. If we use a high-resolution QTOF instrument, we can measure the exact mass of the same ion as 311.0814 with 4 decimal places. We can then put a narrower 3 ppm window around the exact mass. This will result in only 3 possible formulas. It becomes much easier to elucidate the structure of this compound. The exact mass is particularly powerful for E&L testing. According to the revised ISO 10993 Part 18 that you have heard earlier today, any unknown peak above AET needs to be fully characterized. In some cases, you may find many peaks above AET. By reducing the list of possible chemical formula for each peak, the use of high-resolution MS will significantly reduce the workload associated with structural elucidation. Nevertheless, more often than not, measuring the exact mass of the entire molecule alone is not enough. For example, in case of isobaric compounds, with one example shown here, all those compounds have exactly the same chemical formula, C16H22O4, therefore the same exact mass. You will not be able to distinguish them only based on the measurement of the entire molecule. But if you break them apart in the collision cell of the QTOF instrument, they will generate different fragments. The important point we want to make here is the exact mass of fragments will provide crucial information to distinguish isobaric compounds and aid structural elucidation. This being said, let's see how we measure those fragments. Here's the schematic of the Xevo G2XS QTOF showing the main components. Without going into too much technical detail, let's focus on one key element here, the XS collision cell, highlighted in red in the middle of the schematic. Our system has the unique capability called MS to the E to apply alternating low collision and high collision energy, therefore enabling the simultaneous acquisition of precursor and fragment ions throughout the entire analysis. Later, during the data processing, precursor and fragment ions will be precisely aligned based on their retention time and peak shapes. So for every peak, we'll have the exact mass of the compound and its corresponding fragments. Here's an example with Iganox 245, a common antioxidant. During the analytical run, we obtained the mass spectrum of the precursor shown at the top of the window and the one of the fragments shown at the bottom. As you can see, the software also automatically assigns chemical structures to most of the peaks present in both spectra, therefore significantly reduces manual interpretation. Another useful feature of Xevo G2XS QTOF is Unispray, a novel ionization source developed by Waters. On the schematic and the picture shown here, you can see the liquid stream coming out of the column is converted into a nebulized spray and directed at a stainless steel target held at high voltage. Interaction with the target creates a fine spray of charged droplets. They are then directed to the sample cone and enter the mass spectrometer via the ion inlet orifice. In comparison to the traditional electrospray, Unispray provides greater ionization efficiency, therefore better sensitivity. This is particularly useful for trace quantitation during leachable study. Unispray also covers a wider range of compounds with different chemistries and polarities, particularly useful for E&L testing, where we deal with very different polymer additives, impurities, and degradation products. At the same time, Unispray is comparable to electrospray in terms of robustness, linearity, and MS spectra, all the good features that existing MS users appreciate and are familiar with. Here's an example with Uvenil 3030 in positive ion mode. The chromatogram at the top is acquired with Unispray, the one at the bottom with electrospray. Unispray increases the signal by a factor of 2.5 in this case. Here's another example with antioxidant 425 in negative ion mode. The top two chromatograms compare the data obtained with a concentration of 5 ppb on either Unispray or electrospray, with Unispray providing a signal that is 4 times higher. The bottom two chromatograms also compares the two ionization modes, but with a concentration of 0.05 ppb. Once again, the signal is increased by a factor of 4 in favor of Unispray. Overall, the sensitivity improvement with Unispray is compound dependent. But for majority of compounds we have experience with, the average gain varies between 2 to 10. Now moving on to another useful technology, ion mobility, that complements nicely high-resolution mass spectrometry. In essence, ion mobility separates compounds based on their size and shape. When coupled with LC-MS, it provides orthogonal separation, as LC separation is based on retention time and MS based on mass over charge ratio. Ion mobility is useful in many cases. It can separate isomers that even high-resolution mass spectrometer cannot separate, because isomers have exactly the same mass of precursors and oftentimes same fragments. When two compounds coelute or a compound coelutes with a matrix, our mobility can separate them apart, providing cleaner mass spectra, making the elucidation much easier. This can also reduce the number of peaks above AET that need characterization, as the sum of two coeluting peaks may exceed AET, while the individual peak could still be below AET. So separating coeluting peaks upfront could avoid unnecessary work. Last but not least, our mobility can measure the collision cross-section of a compound. CCS is a matrix and system-independent physical-chemical property of the compound. It can be used as additional identification point during screening studies to reduce false positive or false negative. Here's how it works. In this example, the two compounds, the phosphate and Ignox 245, coelute with a retention time around 7.48 minutes. When Xevo G2XX QTOF is used, mixed mass spectra for both precursors and fragments are obtained, corresponding to the sum of two compounds. This makes the elucidation quite challenging. Now, if we use the Vion, a QTOF instrument equipped with ion mobility, to analyze the same sample, the two precursor ions are separated by ion mobility, and fragments of each precursor will also be acquired separately. As a result, we obtain very clean spectra. You can see the precursor and fragment spectra of Ignox are aligned at the left side of the screen, and the spectra of phosphate aligned at the right side. This allows much easier and faster structure elucidation for both compounds. I have talked a lot about the high-resolution MS so far. Now let's change gear and have a look at another critical element of our solution, the informatics. The software we use is called Unify. It is a single software platform for chromatography, mass spectrometry, data management, and lab workflow. It includes data acquisition, processing, visualization, and reporting. It is based on a database structure with configurable compliance tools, so the software can be set up in a fully compliant mode if your lab environment requires so. Another nice thing about Unify is its application-centric workflow. As I will show you in the next couple of slides, the workflow can be easily customized to meet specific E&L needs. Most of E&L testing involves three types of screening. Target screening is carried out when you have a well-defined target list of compounds to look for. This list is often based on the information you obtain from your material suppliers, so you know those compounds have been used in the manufacturing. Analytical standards are available for every compound on that list. Suspect screening is very similar and usually done in combination with targeted screening. This means that you do not know for sure the presence of some compounds, but you have strong suspicion. Analytical standards are available for most of them, but not always. You will use your MS data and screen against a comprehensive library of known compounds that you have developed beforehand. For targeted and suspect screening, the compound identity is confirmed by an identification point such as expected retention time, mass error, isotope fit, found expected fragments, or CCS values if you do use ion mobility. After targeted and suspect screening, if you still have peaks left that are above AET, then you need to continue with untargeted screening. This is really about elucidation of unknown unknowns. As you have no target list, the compound is not in your library, and you need to figure out its structure from scratch. With this being said, on this slide, you can see how Unify workflow is set up to match the E&L testing need and reduce complexity. The typical workflow applies to all compound types where the target, suspect, or non-target. Data is acquired, organized, reviewed in the order shown here at the right side. However, this workflow can be easily customized by yourselves if you want to skip some of the steps or include additional tasks. Let's have a closer look at the Unify workflow. It starts with a wizard-based automatic instrument setup to get the high-resolution MS ready in a time-efficient manner. When the instrument is ready, we can now select the analytical LC-MS method and build a list of targeted or suspect analytes from Unify Scientific Library. You see the example of Igonox 1098 here. For each compound, the library contains critical information such as chemical formula, structure, precursor fragment ions, retention time, and TCS value to allow identification. Waters provides customers with a large E&L library that we have developed as a part of our solution, but customers can create their own library very easily after acquiring and processing data with Pure standards. Now we can start to acquire data in MS to the E mode. As explained earlier, this means after the sample injection, throughout the analysis, we will apply alternating high and low collision energy to acquire simultaneously precursor and fragment ions. In this example, the chromatogram and spectra at the top correspond to precursors. The ones at the bottom correspond to fragment ions. When the run is finished, the data are then processed. Precursor and fragment ions will be precisely aligned based on their retention time and peak shape to provide clean MS spectra for identification. After the data processing, we'll go through the 3 steps of data review, which are targeted and suspect screening, followed by untargeted screening. For targeted screening, the first task is to build a calibration curve for each compound included in the target list. Here you see on the top of the window the calibration table with 4 calibrants at different concentrations. At the bottom, you can see the chromatogram of each calibrant, the integration of each peak, and the calibration curve built from them. After the calibration curve is built, the next step is to calculate the concentration of each sample based on its signal. The example here shows the quantitation results of 6 samples at the top of the window. At the bottom, there is a summary plot. The blue bar represents the concentration of the targeted analyte in each of those 6 samples. This plot allows you to easily compare samples and see how the concentration varies in the whole batch. Moving on to suspect screening. After the processed data is screened against the Unify library, the top of the window will display a list of identified suspects with its identification points. Take the first analyte highlighted in red as an example. Igofox 168 has been reported found with acceptable retention time variation, mass error, isotope match, and expected fragment ions. If you want to see more details for any of those compounds, you simply click on it in the list. The bottom left of the window will show the chromatograms of the precursors, fragments with their retention time, while the bottom right of the window will show their mass spectra. Those information are used to confirm the identity of the peak. After targeted and suspect screening, what else is in my sample? This is the question we need to answer during the last step, which is the untargeted screening. For this, the software will compare the chromatogram of the sample in blue to the one from the blank in red. This will result in the so-called difference plot, shown in green, which shows the unique peaks present in the sample only. If any of those unique peaks are above AET, they will need to be fully characterized. This window shows at the top the list of unknowns that need characterization with their retention time and observed M/Z ratio. From there, Unify will use some very advanced elucidation and discovery tools, such as common fragment search, halogen match, adduct finder, transformation, etc., to propose most likely chemical structures. Similarly to targeted and suspect screening, Unify can also display the trend plot of unknowns, showing the concentration variation in the whole batch. Finally, when the data review is completed, Unify allows to generate fully customized, flexible E&L reports. So far, I have talked a lot about nice features in the instrument and informatics. But I would like to emphasize it is not only about instrumentation and software. There are some really critical elements that make up our total solution. We have developed and optimized a 15-minute-long generic LC screening method using an Acuity Cortex C18 column that offers the highest separation capacity. Based on this method, a Unify E&L library has been developed and made available to all our users. It currently contains more than 700 compounds with MS information, retention time, and CCS values. While we are continuing to expand our own library, as said previously, customers can easily develop their own on the top of it. E&L testing is a challenging application, partly because polymer is everywhere. Consequently, particular care should be taken for sample and system preparation. We recommend the use of our certified low contamination vials, and a specific system cleaning protocol has been developed as well. We also have a system suitability test using a mix of 18 common polymer additives. This should be used routinely to check the readiness of the system before analysis of real samples, and this should also help in case of troubleshooting. Last but certainly not the least, in addition to all those tools, our application chemists have gained deep expertise and experience in this area over the years. They continuously train and support our customers to use our total solution in the most effective manner. With that, I'm done with my presentation. Back to you, Ben.
Now that you've done your E&L analysis, you know what's there, how much is there. So what else can you do with this information? So, amongst other things such as the toxicology assessment of E&L, you may want to know where those E&L come from. They may come from raw materials, the starting ingredients of your materials. However, even if the formulation's starting ingredients are below regulated levels, the processing steps could add non-intentionally added substances, like the NIAS, to the end material via chemical reactions and/or the various stresses that material undergoes during the life cycle. Screening for NIAS, non-targeted, and elucidation can be accomplished using similar methodologies to E&L. You may also want to remove or decrease the level of E&L in the end material, but this is not always easy, straightforward, because in doing so, it may impact some physical properties of the material, such as the rheology behavior, the plasticity, etc. It can potentially impact the processing properties, end-use properties, as well as the lifetime stability of your material. So beyond the E&L end results, understanding the structure-property relationship is critical to ensure the selection of materials with best performance and safety. Waters TA offer a unique, comprehensive suite of technologies that help you in this endeavor. Chromatography, mass spectrometry provide you deep insights on chemical structures, while rheology and thermal analysis are key technologies to evaluate performance, processing, and stability properties. This series will be talking to many of these material properties, how we can help you measure and how we can help you understand your material better. Next week's talk will be on mechanics and strength of biomaterials, presented by Troy Nickel. We hope to see you then. We will now take some time for questions. We have with us Dr. Caitlin DaCosta. Caitlin works with our customer success team as a senior application scientist for the UK and Ireland regions. Caitlin covers pre- and post-sales application support for both high-resolution and tandem quadrupole mass spectrometers. Her focus is in the chemical materials market and extractables and leachables applications. Whilst we collate the questions that you've been submitting, we'd appreciate if you could answer a short poll to help us prioritise how we can help you better.
Okay, so we have our first question here. So thank you for phoning the poll. Our first question is how to do the quantitation in untargeted screening with MS. Caitlin, I think this one's directed to you.
Hi, Ben. Thanks. Yeah, so what we typically see in the field is a semi-quantitative approach for unknowns using a standard. So this standard is normally spiked either into the sample or analysed separately. And the concentration of the standard is generally at the analytical evaluation threshold, or AET. And we can use this response from the standard as a threshold measure to identify unknowns above that response. From here, it's possible to use the tools that we have within the software to help us elucidate or identify these unknowns, and then we could quantify them in a targeted manner.
Okay. Another question here on a workflow question. So the flexibility of workflows and scientific library is quite important. Can you explain how you developed, optimize your LC-MS methodology and the related DNL library? Oh, sorry, just before I answer that one, there's a follow-up question to the previous one on the untargeted screening. The intensities can be very different of standards compared to unknowns, especially with MS. Could you quickly comment on that one?
I mean, yes, of course, and that is true, especially with electrospray ionization, just as an ionization technique. Different compounds will ionize with different efficiencies, and therefore you will get different response. Typically, we try to take that into account by using a 50% margin. So where your AET is, we would, or quite often we see people dropping the threshold to 50% of that to try to take into account the different ionization efficiencies with electrospray. It's a very good point though, and it is something to be aware of. That's why we would typically call it a semi-quantitative approach in this manner.
Okay. So the next question was, let's go back to it. So the flexibility of workflows and scientific library is quite important. Can you explain how you developed, optimize your LC methodologies and the related DNL library?
Of course. So we've been doing a lot of work at Waters around developing a library and a related LC method to cover a range of compounds associated with extractable and leachable studies. Because it's a screening approach, the LC-MS method is it's quite a broad method. So we're targeting a broad range of different compounds using quite generic conditions that help promote the ionization of a range of different compounds. The focus of the library is really around the quality of the library, because this really helps us improve confidence in the identifications we make when using a library to screen against. So we try to include multiple identification parameters such as retention time, structure, experimentally derived fragments, and ion mobility measurements if we're using the Vion and ion mobility as well. If you are interested in more details about our LC-MS method and associated library, please reach out in the chat and we can provide some more information.
Okay, I've got another. Caitlin, I think this is one for you again. You touched upon the use of the Vion and ion mobility. Could you please explain a bit more?
Of course. So the Vion brings the benefits of ion mobility into a high-resolution, accurate mass platform that's been designed really for routine screening. So ion mobility, just to recap, separates the ions in the gas phase based upon their size, shape, and charge. And from this, we calculate a unique collisional cross-section measurement for each ion. We can then use this CCS, or collisional cross-section measurement, as part of our screening protocol as an identification parameter. So it's similar to retention time and accurate mass, and this can help us enhance our confidence in that identification. Also, because ion mobility is a gas phase separation, the CCS measurement is not impacted by changes to chromatographic conditions or matrix. It acts almost as an orthogonal separation mechanism, which to our LC, which again, as I said, can help with identification, but also with spectral cleanup. There's multiple advantages of ion mobility.
Okay. We've had a couple of people asking about the presentation, if it's possible to get it after the event. So the recording's going to be made available, and we'll also make the PDF available as well. So we'll provide you the links to those. So there's a question here on, okay, so we're familiar with food packaging, but are interested in medical device applications. Okay. Would some of our compliance testing for food contact materials be of any use, or would we simply go to the ISO 10993 testing? Okay, so I'll take this one to give you a quick break, okay? So, well, you're certainly in a very good position there. So food packaging is a little bit more biased towards like targeted screening. But you still need to do things like, elucidation of the non-intentionally added substances. So you're very likely, well-versed in technology and the overall process. But obviously, you just need to read through the ISO 10993, especially Part 18, to ensure that you have the correct, extraction methodologies. Of course, the analytical evaluation threshold, the AET, is critical. But yeah, you're certainly in a strong position there in terms of your existing knowledge. You just need to apply the methodologies as stated within the ISO 10993. Hopefully that answers that one. Okay, we have another question here. Okay. Oh, so this is following on from your comments about the library. So how do you create a library entry if the compound of interest contains several key substances in a specific ratio and not only a single one? Can you do one single entry that considers retention time, mass, and the respective ratios? Caitlin.
So typically, when we're creating a library entry, we can either do this using an Excel spreadsheet template, or we can do it based really around a mole file to start off with that we supplement with experimentally derived information. So, for example, the retention times and the fragmentation or known fragments for that compound. And just rereading the question, if the compound of interest contains several key substances in a specific ratio and not only one, how do we create a library entry? The way I would probably approach that is to create a library entry for each of the substances, and then we can use custom calculations to look at those ratios between the different library entries. What I would say here is we do have a team at Waters dedicated to helping customers develop their libraries. We work in collaboration with a lot of customers to build our own libraries, but we also do work with customers to help them generate their own. So if you would like some more information around that, please reach out and we can put you in contact with the library development team.
Excellent. Thank you, Caitlin. Another question, which is certainly in your wheelhouse again, I'm afraid. This is about the MSE acquisition mode that was mentioned for this analysis. So the question is the benefit of this over the other approaches such as DDA.
Yeah, so as we've already mentioned, MSE is a data-independent acquisition mode. So all of the ions that enter the mass spec are exposed to a low energy in the collision cell, which creates our MS precursor ions, and a high energy generating our MS fragment ions. So unlike other techniques such as DDA, MS to the E is a completely unbiased approach in that we do not have any pre-selection of our ions for fragmentation. So we get structural information for all of the ions, even if they're targeted or not. So for both our identified and also our unknown ions, irrelevant of their response. DDA and similar techniques to that typically use an intensity threshold or an approach such as the selection of the top 10 ions for MS-MS. So they're targeting the ions for MS-MS. These ions, or the most intense ions, are not always the most important compounds in the sample. As we've touched upon, different compounds can ionize with different efficiencies. So MS to the E and unbiased acquisition modes really give us a wealth of information when we're screening and we don't always know what's present in the sample.
Okay, thank you. Still scrolling through these questions. So there's a very good question here. So since different compounds have different levels, as we've already discussed, even though it has the same concentration, how can the line be set, so the AET? I would say that compounds below this line is not important to identify when in fact it's possible that a compound just have low response. Very good question. Does anybody want this on the line? Did I have a crack at this one? So, I mean, I think this is a critical question. And it speaks to a few different areas. So one is the importance of understanding what potential contaminants and compounds are in your material, so that you can select the correct technology and the correct ionization source, for example, on how to detect these, and also that helps you set your AET if you understand the expectations around the responses of your material. So this is where working with the value chain is so important, and this is why this is so difficult to do if you have zero information, because as you say, it is perfectly reasonable that you may miss some things because it's got an extremely low response. That's why you tend to have a lot of techniques which cover the breadth of volatilities and material types, and why you need to really have a good understanding of what your material is. So yeah, I think that's one of the key questions and the key reasons that these regulations are being looked at and updated. Does anyone else have further comments from our side?
Yes, Ben. If I may, I will just add one point. I think now, as Caitlin mentioned earlier, we are developing and expanding our ENDL library. And there's one more factor that we are measuring, which is the relative response factor for each compound versus the standard that we will be using for the to set the AET. So I think this is something we see more and more customers start to do when they create their own library. So they will always measure, I think, the relative response factor versus their standard. That will give them actually, I would say, a kind of margin in the future when they set the AET value, because if they look at their whole library, and they will get a good feeling about, you know, the response factors of different compounds versus the internal standards. So I think that will, in the future, help people set the better AET, taking those differences in response factors into consideration.
Excellent point. Thank you, Jink. Okay, so how are we doing for time? Okay, so we better start to wrap this up. So I have a question from Caitlin. We have covered this slightly in one of the comments earlier, but there's someone very new to this area, and this understandably it looks quite a complicated area to move into. Could you please explain what help they would expect to get from us to help them be successful?
Of course.
I don't know who wants to take that one. Caitlin.
Yeah. So, of course, I mean, we understand that this is a complex challenge for all of our customers. So we really are dedicated to providing the appropriate training and support that we can to help them be successful with these types of challenges. So we have dedicated training and support plans with a range of different colleagues of mine, where we will, guide, train the customers in how to use the hardware, how to use the software, but also share our knowledge with what we see occurring in the field. And so, for example, how we see other customers handling some of the challenges that they may come across. And moving on from the training plan that we have ongoing support networks for our customers. And we are continuously looking to improve and learn as well as a company. So we often, as we've touched upon now, are looking at how we can develop our libraries and our methodologies to help support customers, taking into account where we can, the regulations and the changes in the regulations that our customers are facing. So it's very much an ongoing relationship between us and our customers to overcome these challenges.
Okay, well, I think with the time hand, I'd like to wrap this up. Thank you, everyone, for attending this webinar. This is part of a series, so there's still plenty of time to register for the other talks within the series. I'd like to thank Jing and Caitlin for their attendance today and answering all those questions for us. And I hope to see you online again soon. Thank you very much.
Thank you, everyone.
Thank you.
Bye for now.